Good day, this is Professor Will, CBMD PhD. Today's Wednesday, October 21st, 2020. It is 4:01 and 32 seconds a.m. Eastern Standard Time, and this is a continuation of my microbiological research. So let's hop to it here. The nucleic acid probe has to be labeled prior to hybridization in order to detect the form hybrids. The label could be covalently bound to the probe direct labeling, or an unlabeled detector group can be covalently linked to the probe and detected by the labeled binding protein. Indirect labeling. Nucleic acid probes have broad applications. They can be used not only for the detection and identification of bacteria, but also for the diagnosis of infectious disease, the detection of pathogenic and spoilage bacteria in food products, and for studying microbial ecosystems. Nucleic acid probes are highly specific, and their genomic targets are not affected by changing environmental or physiological conditions. The technique is faster than conventional identification methods, and can be applied in the identification of organisms grown in pure or mixed cultures. Nucleic acid probes are especially useful for the identification of slowing and fastidious organisms. They can also be used for the direct detection of bacteria in their natural habitat. Even bacteria that have not yet been cultivated can be detected either through the use of reverse transcriptase sequencing of extracted rRNA, or by applying the polymerase chain reaction with oligonucleotide primers that are complementary to conserved regions of 16 or 23S rRNAs. A major disadvantage of the nucleic acid probe technique has been its lack of sensitivity. However, this problem can now be solved since there are several ways to increase sensitivity, e.g., amplification of target nucleic acids or signals using high copy number target sequences or improved detector groups. Amplification of target nucleic acids with polymerase chain reaction has considerably improved the sensitivity. This technique was used to detect as few as 35 colony-forming units of L. pneumophilia in a water sample. Physiological properties of bacteria are expressions of the metabolic pathways present in the respective species. As far as types of overall energy metabolism are concerned, the relation to oxygen and light of a given bacterium will decide whether it performs photosynthesis, fermentation, or respiration as the main energy-yielding process. The ability to utilize light as a sole source of energy clearly separates the phototrophic bacteria, including the cyanobacteria, from all other prokaryotes. Of course, this ability requires the presence of the numerous properties that constitute a functioning photosynthetic apparatus. In addition, a large number of bacteria have the ability to grow in the absence of its oxygen, as well as in its presence. These facultatively anaerobic bacteria poses more than one energy-yielding system. Metabolic properties of a given bacterium are detected and determined by studying the utilization of nutrients, the production of metabolites, or the presence of typical enzymes. In diagnostic bacteriology, especially in the medical and food sectors, such specific biochemical tests are routinely performed for the identification of bacteria. The fermentation of glucose, lactose, mannitol, or other sugars may be easily detected by the production of gas, or by changing color of an indicator of dye caused by the production of acids. The qualitative and quantitative analysis of acid end products is an important aid in the identification of bacteria, especially in the anaerobic bacteria of the genera Clostridium bacteroides, Peptococcus, and Peptostreptococcus. Volatile fatty acids may be estimated in the culture suspension, in an ether extract of the culture, or in the bacterial cell directly or after derivative formation, by gas-liquid chromatography, or by other techniques such as isotachophoresis. In addition to the acid end products such as presence in the medium of other fermentation breaking down products, such as indole and hydrogen sulfide, is characteristic for certain bacteria. Thus, the ability of an organism to produce indole by enzymatic cleavage of an amino acid tryptophan has long been used as part of the IMVIC reactions used to separate Escherichia coli from the Enterobacter aerogenes. Good day, this is Professor Will, CBMD PhD, on microbiology, bioinformatics, and computer science. We hope you enjoyed listening. Until next time, have a great day.